holidays next week for those. So that also kind of helps, I think, free the schedule up for those who are trying to balance that. Okay. All right. So today, my pleasure to introduce, do you go Matthew or Matt?
Let's go, Matt.
Okay, Matt Pennell, from QCB, okay, Comp Bio over the main campus. And it's been here just for 2 months.
2 months, yeah.
So,
Now I'll give some of my background.
oh, okay.
Yeah.
So, yeah, I'm gonna shut up then. And so, title of the talk, go.
All right. Thank you very much. Thank you very much. All right. So appreciate you being inviting me here. I'm over at QCB and also in biological sciences over the main campus. I just got here in August, but before that, I was a associate professor at University of British Columbia in Vancouver, and I just came over here. And so I'm kind of maybe a bit different than most of your speakers. I'm an evolutionary biologist by training, and my first bit of my talk is going to be quote unquote pure evolutionary biology. So if you're really bored of it, just close your eyes or something, but it's kind of setting up a story, which I'm going to talk to you about epidemiology and why I think this is the work I'm doing is relevant to the problems of genetic epidemiology. I'm going to be talking about one kind of part of my lab, but there's kind of 2 other portions which I'm not going to talk about at all today, and I'm happy to chit chat with people. I have 3 people working my students Ciel, postdocs Alex and Rex all work on kind of evolution of gene expression across species and maybe how that co-evolved with protein abundance, etc. I also have 3 graduate students who are all working on immunoglobulin diversity and how that arises. I'd be happy to chat about those projects. But that's not what I'm talking about today. What I'm going to talk to you about is this really weird rabbit hole I fell down for 3 or 4 years. And I'm just now coming out of the other end of it. And produced some very, very, to me, surprising and striking results, which have really fundamentally altered the way I think about these problems, but also science in general. Okay, so like I said, the first bit's going to be pure evolutionary biology, but it's going to set up the problem. Then I'm going to apply those to epidemiology and show you what I'm doing with this. I think because the epidemiology doesn't make sense without the rest of it. Okay, so this is kind of my stick here, basically looking at macroevolutionary patterns, building tree of life, and figuring out what we can learn about this. One thing that lots of people have documented in many different groups is that some species, some studies of organisms are much more diverse than others. And ultimately, these speciators, other groups seem to have slow extinction rates, etc. And these are kind of used to explain disparities among taxa. We also know that species diversity varies a lot per regions. Okay? And this is kind of looking at North American versus South American bird groups. And basically, you can see that the pattern is completely different. Species accumulate a lot more with time in South America than they do in North America. So kind of these regional disparities in speciation extinction rates, really important. And paleobiologists have been talking forever about modeling speciation extinction rates through time and kind of figuring out why diversities are risen and fallen over the past history of life. So that's basically the summary of the research problem. Then I'm going to research program. Then I'm going to kind of talk to you about the how we might ask these questions, which is going to lead us into these problems about epidemiology. So I talked about this kind of process of speciation extinction events. So how are we learning about the rates? And in particular, our best information for most groups is only contained in the phylogenetic tree of extant organisms. We don't have genetic data or very much data for many fossil groups at all. And so we're going to try and make use of the phylogenies of extant critters to kind of infer historical patterns. Now, the first side to the process, and at the end to the present, we sample these guys. But the ones we've sampled are only a subset of the whole distribution of lineages that ever existed. So when we reconstruct the phylogeny here, we get a tree that looks like this, which is different than the real tree here. But we can use the distribution of branch length through time to get an estimate of the rates given some model. And again, this tree was derived under a birth-death process where the birth rates, splitting events occur at some fixed rate, and the extinction events occur at some other fixed rate. And a simple way to think about how we get information on these parameters is if you imagine the speciation rate lambda, extinction rate mu. If we plot the number of lineages through time on a log plot right here, what we see is that they expand linearly through time. Curve is different is because things which are around today had not had the opportunity to go extinct. So basically, we can, judging by the branch length of the distribution of the tree, infer these parameters. And this is really important because different types of models make different predictions about the distribution of branch lengths through time. Okay, and an incredibly large research enterprise in evolutionary biology is coming up with different hypotheses for why diversification rates may have changed, maybe depending on the group, maybe depending on some feature of the group, maybe depending on the location, maybe depending on some environmental kind of situation, and coming up with different kind of models. And we can, each one of these models make different predictions about the distribution of branch lengths through time. Okay. And like I said, this is like an entire huge research program in evolutionary biology that I have contributed something to. So that's kind of the setup of the problem. Okay, that's interesting. But I'm going to say that the evidence we've been using in this kind of field is probably not the right evidence. Okay. And I got in this deep rabbit hole with my a former postdoc, Selena Luca, who's now a professor at University of Oregon. And we went to, like, in a roundabout way, we got to tackling these two related questions. First of all, so I said there's all these models that produce different distribution of branch length. Which ones can we tell apart with phylogenetic data? And how robust are these parameter estimates for these models if we misspecify even a little the actual differing process? We all know we make simplifications in biology. That's not news to anyone. But we want to know how robust our estimates are to misspecifying the process. Okay. And there's been a lot of work in this area in evolution, basically doing various simulations. You can imagine what they are, like simulate under this model, fit this other model, see what answer you get, et cetera, et cetera, et cetera. Look at how large the data you need to infer these parameters as well, under given assumptions, et cetera. And we've also had case studies where people compare the estimated speciation and extinction rates from molecular phylogenies to what they observe from the fossil record for different groups for which there is good data available, such as the cetaceans, the whales, and the dolphins. But this is kind of unsatisfying because these are all just case studies and sometimes it seems they work well, other times it seems they don't work well. So what we started doing here, we're trying to think about, let's deconstruct this problem to its very core. Let's try and figure out in abstract, any kind of birth death model of speciation extinction we can come up with, let's kind of create a general formula for studying this problem. So we're going to consider here a birth death model, where things lineages split at some birth rate lambda and go extinct at some vary, for instance, linearly through time. That's a fine function. We can also have, of course, constant through time. Whatever. So cases in a second, when all the lineages are sampled in the present day, there's well-established formula for expressing the likelihood of that phylogenetic tree given that model parameters. So whatever we want to do, we can always express the likelihood here. And this is my slide that probably isn't relevant for the people in this audience, but it's part of my standard repertoire talk. But basically, as you should know, the likelihoods have a measure of surprise. How surprised should we be given a set of parameters to observe this exact tree? And we can use the likelihood to estimate the maximum likelihood parameters, do Bayesian inference with MCMC, get the posterior distributions for all the parameters of interest. And we can also do model comparison using standard approaches like AIC, BIC, Bayes factors, and what have you. So that's good. But basically what we figured out was there's an alternative way to write the likelihood that people have been using before. And this is a pretty boring technical detail, and we published, originally we're thinking a pretty boring technical paper. And the reason we were actually kind of looking at this, to be honest, was we're trying to speed up some calculations to do some really big analyses. Now, it turned out we could write the likelihood of any diversification model directly from the curvature of this deterministic lineage through time plot. So I say deterministic because it's every time we do a sampling of some kind of birth death process, there's lots of variance, of course, but in expectation, how many lineages do you expect to see at every time point? And you can actually compute this expected deterministic lineage through time plot given any model. Okay, and this is the deterministic lineage through time plot for a birth death model, but you can actually write the likelihood of any one of these complex variations just using the implied curvature. And because the likelihood only depends on this deterministic lineage through time plot, any two models that have the same deterministic lineage through time plot have identical likelihoods and cannot be distinguished with any data set, no matter how large. And we call these models, such models, congruent. Okay, and we call them congruent because it's like a congruent triangle. In fact, I'll show you in 1 second. There's actually a geometrical explanation to all of this stuff I'm going to talk about. Okay, so if we now know that any two models that have the same expected curvature of the lineage through time plot will have the same likelihood, well, this actually gives us, we can actually kind of back calculate and use this tool to find other combinations of parameter values that generate the exact same distribution. So it was kind of known before, to some degree, that different processes could lead to similar distributions, but we basically solved a way of finding the exact one for any kind of arbitrarily complex birth death process. Okay, and so basically what you know here is that here's our lambda and mu, which can be time variable, so any given time point. We can actually have the curvature of this thing called the fold speciation rate, R tilde. It's going to come up again. Has starting conditions, and basically the tilde at any given point is the speciation rate at that point minus the extinction rate plus the inverse of speciation times the derivative of speciation. So this is a pretty meaningless parameter here. It's a composite of all these, but it turns out that this thing through time contains all the information one needs, in addition to the starting conditions here, to calculate a likelihood. So because that's true, any two models that have the same for any given a set of lambda and mu, you just can insert a new mu and solve for the corresponding lambda. Okay, so this gives us a technique for finding models that have the same branching distribution as our target model. Now we can go hunting for these. Okay, so it turns out, in a really wild surprise, that the models that have the same likelihood can have totally different dynamics. Okay, so here's 4 different models: dark blue, light blue, pinkish, red solid line. This one's got a speciation rate that goes like this, and extinction rate peaks here, extinction rate peaks here, or maybe one that goes here, one that's constant. And this is the difference between these two, all variable. These have all the exact same distribution of branching densities through time and cannot be distinguished with any data set, no matter how large. Now, this is very disturbing because these things are very quantitatively different and qualitatively different, and are different enough that, like, these are hypotheses that people are trying to tell apart in the literature. Was there a peak in extinction here or here? Has extinction rate declined, or has it been constant? We can always find these large sets of very qualitatively different models, which we call congruent sets, and they're actually form a geometric group. Okay, and these are just geometric transformations of these functions, and they all result in the exact same likelihood for any tree and cannot be distinguished. Okay, and then you get this thing that kind of goes wild, where you have like the biggest tree we have for is angiosperms. There's like 80,000 plants with genome sequences. We build a tree there, and we can say, okay, there's a declining speciation. There's one model which has declining speciation and declining extinction. Another model which has increasing speciation, increasing extinction. Both those have identical likelihoods, cannot be told apart with any data. And this is critical because people are doing, including me, are interested in, are these angiosperms expanding or declining? And both of these seem to be true at the same time. And this paper the pieces of this situation. Yeah, sure.
Is this problem get better if you specify more complicated starting models?
No. No matter how complicated your starting model, I can always find very different solutions, no matter what. Now I'll show you in the case of epidemiology where this kind of comes into play too. Okay. The way I think about this conceptually, and this has helped me a bit, it doesn't help you, but if you imagine yourself like this pink blob is a two-dimensional shape, but just imagine in your mind's eye an infinitely dimensional hyperspace. Okay. And imagine, that's kind of a joke, and not that funny, but anyways, can't really imagine it, hard to picture it. But if you do a model comparison, and in this case you have 2 models you're comparing. Now, here's your true generating process. Okay. That they gave rise to the tree. Here's the model in your set, which is closest in some geometric sense to your true generating process. But this model is closest to a model in the same congruent class as your model you look for. The generated tree. Well, this one's going to be favored here by any kind of model selection procedure. And this is also true if you have a model of one of your sets of models that are actually in the same congruence class. You're going to have strong support for that model, even though it's very different mechanistically than the model you actually generated the process tree. So that's a pretty stark conclusion. It's impossible to reliably estimate these diversification rates without other information. We just cannot do what we've been doing for the last 30 years in this evolutionary biology. We don't have enough information to make the claims we want to make. Now, a lot of people, when we publish these results, they were skeptical, not of the math itself, but skeptical that this actually mattered in practice. Well, sure, alternative models might explain data, but that doesn't mean our data, our inferences have been misled. And I'm going to show you that this have been misled this entire time. So, a weird phenomenon in molecular phylogenetics that happens all the time is that estimates for molecular trees for extinction rates are often zero, or very close to zero. And this has been a long during a tree, estimated zero extinction rates for this group in 2014. But we know this is kind of in some level dumb because we know there's extinct hummingbirds. Okay, so something's wrong with here, but it's never been really clear why this is the case. And there's been lots of hand-waving explanations. Well, people said, oh, it could be model inaccuracy. Like, I mean, people meaning me as well. Okay, it could be model inaccuracy. Maybe we're missing something here. This could be due to phylogenetic error. Maybe we're like missing all these explanations were always so unsatisfactory for so long because there's no reason that any of these should lead to an inflated estimate of zero extinction rate mathematically. Okay, but our kind of way of thinking about this problem actually does suggest an explanation. Because these congruent sets of models are defined by geometry, not by biology, we know extinction rates have to be positive, but the geometry doesn't know that. And there's no reason that there couldn't be support for a negative extinction rate model. And so all of our programs for estimating these kind of approaches, and like I'm not blaming anyone, I mean, like I wrote the programs most people use to estimate these kind of things, is we stop here. And see what happens. And it turns out that for the hummingbirds at least, the best maximum likelihood estimate once you turn off this flag is the extinction rates are negative. And so our hypothesis here is that why we see these little results is that models estimates are converging to a model with a negative extinction rate. But because we're putting this boundary in this program, because we're smart and we know about biology, it's just piling up on this boundary condition. And we're seeing this over and over and over again for 30 years without figuring out what was happening. And so this, yeah.
What is the data that's driving this negative?
The reason it's going to lie here, I can go back and show you this right here, is because we're in a situation like this. Okay, so this is the true generating process here. And this set here contains models with a negative extinction rate. And so if you happen to compare models that are just off this here, and this one's closest, it's going to convert to a negative extinction rate. That's our claim. And this is going to make basically several predictions here. So, first of all, we should see the extinction rates equal zero are possible, even when the models are adequate, even when they have completely described the data in reasonable ways. If extinction rates are positive, it shouldn't be affected by the boundary being removed. If we fix lambda to its true value, we essentially collapse the congruence class to a single model, and therefore the estimated extinction rate should go to its true value, a positive number. And even when we estimate, our thing predicts that even when we estimate the extinction rate is less than zero, the estimates of this R tilde should match the generating model. That is to say, if we know it in the simulation, if we know the generating model here, the R tilde for every time point, the negative model we find should match that R tilde. And basically, how we did this with some simulations and some data analysis. For simulations, we simulated these very large trees. They landed in mean vary through time. We fit a bunch of different models. We picked the best model using AIC, threw out poor fitting models, and then we just altered this boundary condition. And all of these kind of conditions are rates, this boundary is imposed, you do the pilot in this boundary, and when we relax this condition, they all fall below. The positive ones also stay positive. When the boundary's there or the boundary's not there, the positive ones all stay pretty much what they were. And if we collapse congruence class by fixing lambda to the true value, all the estimated mu's are lined up really well with their the true mu's with some sampling errors, etc., and optimization errors up here. Of the negative extinction model is basically very, very, very close to the R tilde for the true generative process. It's working. These processes are working. And I'm just going to skip this a little bit because it's not that important. But basically, for real data, we can't know the real truth, of course, but we can still do this kind of altering the boundary experiment, the condition experiment. And what we see here is similar patterns to what we observe in the simulated data. When extinctions fix at 0, there's a path in the boundary, and they all go below 0. When extinctions positive, they basically resemble their previous value. So all this goes to support that, like this observation that the extinction rates have been 0, it seems to be consistent with our model congruency explanation. And we actually have a real mechanistic explanation of why this can happen. And if these extinction rates are 0 because of this model congruency, not because of some kind of phylogenetic error or something like that, it means the speciation functions must also be therefore involved. So this is saying that these estimates for 30 years seem to be wrong in the exact way for the reasons we've described in our article. An actual, real empirical phenomenon. So my background, as I said, is in phylogenic trees. We study this process of species diversification, where we have tip samples of the present. But when we're looking at viral epidemiology, what we're doing is we're using viral phylogenies in which tips have been sampled throughout the process. Here's a aspect to this too. And these are the models we use for biophylogenetics are very similar to the ones we use for speciation and extinction, but they're different by this fact that the data is collected through time and not sampled at the present. So you think, okay, maybe that's more information we can get that can allow us to distinguish different scenarios. In the case of viruses, and looking at viral phylodynamics. And this is with Eileen McPherson, who's now probably Center for Fraser Math, and she was at my postdoc at UBC. And then Jeff Joy and Angela McPherson are both at UBC Medicine and the BC Center for Excellence in HIV and AIDS. And they are, in addition to academic responsibilities, really involved in setting the HIV policy for British Columbia. So, okay, let's go why this whole problem is related to epidemiology. So, just gonna, obviously this is not that interesting to you all. We have an SIR model. statistical kind of mathematical modeling, looking at whether this infection will increase or actually looking at all the individuals. And so for many emerging, understudied, and undersampled infectious diseases, models like this can be often difficult to parameterize. And as such, it's been a huge motivation over the past decade or two to use phylogenetic trees of the virus to parameterize these models. And in fact, you can use the birth, treating birth here as a transmission event of the virus, a extinction being a lineage extinction in a host, a recovered host, and then we also have these kind of sampling parameters here, a sampling of the viral lineages. And so we can, at least conceptually, estimate the parameters of some kind of compartmental epidemiological model from the branching pattern of a phylogenetic tree reconstructed from the viral samples of different individuals. And this has been done a lot. I just picked some random example, looking at HIV in the UK and charting effective reproductive through time under the simple SIR model. You can do this with other different types of models, whatever. But basically, this is becoming like a major toolkit in the epidemiology kind of world to combine it with traditional compartmental models, especially for undersampled diseases. This has been proven to be pretty useful, and they're using these techniques in British Columbia right now to study HIV and policy. So this is not a fiction. This is widely being kind of used in terms of the actual people who are doing epidemiology. So we wanted to ask the question, does our kind of way of thinking of this problem relate or have any kind of influence on these kind of research programs? And like, what does that mean for estimates of viral phylogenetics from these trees? So we want to try and this is a two-wordy slide, and I apologize, I'll just explain it. So we wanted to try and figure out how to extend our results from macroevolution to the space of viral epidemiology. But there's a big technical hurdle that remains that's pretty not that interesting to most people, but it's very interesting to us, is that for any given birth-death model, which we sample all the lineages at the present, we can compute a likelihood pretty easily. We knew there's known formulas, but there's not a known formula for calculating the likelihoods of speciation extinction rates through time for any arbitrarily complex model given sampling through time. Okay. So I'll just skip the point here and get to the rub. So basically, in order to start studying this problem, we have to create a generalized likelihood framework for actually calculating likelihoods for any arbitrarily complex tree. And my postdoc, Eileen McPherson, basically solved this and created this formalism for deriving tree likelihoods under arbitrarily complex processes. So, okay. So we were then able to extend the results from the macroevolutionary case to the epidemiological case. And we can, rather than just defining these models by their branching density, we can define them as a combination of the expected branching density and the expected sampling density. And when we do this, we can apply the same kinds of tricks we used in macroevolution and show that these dynamics still come into play even in this case. That these are, like in the macroevolution case, 5 different models, which have different transmission rates, different lineage extinction rates, different sampling rates, and very different R rates, but they all have identical likelihoods. We can never tell these apart, no matter how much we try, no matter how many data that we have, no matter how many samples we have, never tell these apart. Okay, so we can basically extend the results from macroevolution to this case of the viral epidemiology. And like I said here, so this is really important because, R equals 1. This is a critical barrier. Is the disease spreading or not? When we get things that are way above 1 and quite far below 1 are exactly equivalent in terms of the model support. Now, I'm going to show some really fun results to show this actually matters a lot. So hypothetically, this could matter actually, because if we fit some model to some data, we can now start to explore, just as we did with the macroevolutionary case, the alternate scenarios. This is from an HIV epidemic in Alberta, Canada. This is real data from a real public health study, and they had basically estimated these parameters. The dark line here is the median estimate. These darker line shaded areas, the 50% credible interval, and this is 95% credible interval. Right here, this is using the software Beast, which is the standard in this field. Okay. And what we can just basically do is take their results and just apply our transformations to it, and we can get all sorts of wild different epidemic trajectories here. So it could work out, but we want to explore this a bit more with simulations, because you could say, well, sure, there could be multiple explanations for this given data set, but does it actually affect inference? Now, in the macroevolutionary case, we kind of made the case that this did, owing to our effect on the results of the zero extinction rates, but we're going to try and see if this cashes out in the epidemiological case. So again, we have our blue blob replace our pink blob, because we published this in a different paper. We couldn't reproduce the figure. We have to change it a bit. So what we'd like to do here is that the true pattern of transition rate changes through time, something like this, and we're going to fit some piecewise constant or piecewise linear kind of model to the data, which seems totally reasonable. We should hope that it approximates the true value. We're never going to get the continuous function exactly right. And maybe it's not even continuous, maybe it's truly discrete, but we should be able to approximate it with a model where it's allowed to change at discrete intervals through time and have like a piecewise constant or piecewise linear model. So that's the ideal outcome. But I'm going to show you we do not get that. Okay, so what we did is we divided our team into two. Okay? Half the team did simulations, the other half did inferences. And they were totally blinded to each other. And I was on the team simulation, and the optimistic HIV biologists were on team inference. Okay? So what we did, and we did this many times, the case is always exactly the same, basically. We simulated parameters here. Okay. In this case, effective reproductive number, this is a delta S, doesn't really matter. They're just different parameterization of the same model. And this is the simulated process, nothing too complicated. And this is the inference that the methods got. Okay. And here, effective reproductive number was straight. And the inference team was estimating that it was up here, then right rose, and came down here. Okay. And we see basically hardly any resemblance at all to the true dynamics. Again, this is a different parameterization, doesn't really matter what the difference is, called removal rate. This is what it looks like. And for the entire history, it's outside the 95% credible interval for this true problem. So, okay. And we fixed a bunch of other parameters like the clock parameter, all we got all of the other things right. So why the heck is this going on? Well, one thing you can think of is maybe Beast 2, there's an error in this problem. Maybe it's not working properly. Maybe it's not doing what we think it should be doing. That's why we're getting these really weird results. But we can test that. What we can do here is we can convert this model right here that we have here into its like what do we call these composite parameters. This deterministic branching density and deterministic sampling density. This is the equivalent of R tilde for the case of the sampling through time. Okay, and so these blue lines right here are the true model, and these right here are these models projected into this space. So what this is telling you is this is a really, really good estimate, okay? And this is a really, really good estimate. That means BEAST, which is the standard software for doing viral epidemiology, is nailing the congruence class of the model. It's getting the model, like, basically in the limits of finite sample size, almost perfectly. It's performing exactly as we think it should, but it's converging to a model which is in the same congruence class, but it's qualitatively different than the true generating process. And we see this over and over and over again. We do maximum likelihood, we do Bayesian, we do all sorts of priors. We get the same result over and over and over again. It's always converging to the congruence class, but not to the correct dynamics together. And basically, if you fix one of the parameters, you collapse the congruence class, and you can recover the, basically, if you fix one of the parameters here, you can collapse the congruence class and recover the correct dynamics more or less. So it's all exactly kind of performing as advertised, but also subject to the pathologies that we discovered. So basically, if this is the true dynamics, we're recovering models oftentimes that look like this. They have basically no correspondence to the true dynamics, but they're identical to the dynamics of the congruence class, or more or less. Okay, so basically, and it doesn't matter about putting priors in the parameters, you still get this result. It doesn't matter how informed the priors are, it still cashes out like this. We're still trying to figure out, and there's actually a paper that not by me, but just a number of people started working on this problem. Basically, I'm reviewing the paper. I shouldn't say that, but okay, we're basically exploring conditions and when you expect it to be closer or less far, or why this happens exactly here. And it's kind of a weird geometry puzzle, but nonetheless, it affects inference. Okay. So what are we doing about this? Well, my group is working on 4 possible solutions, not mutually exclusive. The first one is, okay, we know we can't infer these parameters reliably from the phylogenetic tree alone. And in fact, combining them with simple case data is actually not helpful either, because if a positive case is basically just a tip that you don't know where it connects to the phylogeny. So if you knew the phylogeny perfectly, you solve this problem. You definitely have this problem even if you can't know where a sample connects to the tree. But we're working on combining other types of surveillance and clinical data, especially negative test rates and duration of illness and these kind of other observations, and joining them together with phylogenetic trees to basically resolve these congruence classes and figure out what the actual viral dynamics are. We're also working we have created this general approach for deriving tree likelihoods. And for the first time, we can actually derive a tree likelihood under any given compartmental model. So you can give me any kind of complicated compartmental you want that's well built for COVID or HIV or whatever, and we can actually calculate tree likelihoods under that. And what we're working on right now is kind of introducing mechanistic constraints to say that the dynamics have to be consistent both with the tree topology and the supposed mechanism that leads to this data. Also, it's pretty kind of interesting factoid is most of the data we get in this field for viral phylogenies is, I'll just go back here, is basically opportunistic sampling. Someone gets sick, they go to the clinic, they get swabbed, and that gets deposited sequence, deposited in our database. That's opportunistic sampling. And the problem with opportunistic sampling is that sampling rates also change through time as a function of all sorts of things. But if you actually sample differently and actually have large scale concentrated sampling events, this is shown here from Wuhan, China, and there's been a bunch of, especially in China, a bunch of other similar examples where they've done large surveillance testing of a given location in a short duration. This kind of sampling strategy actually can provide additional information that can distinguish these alternate scenarios and kind of allow us to recover the dynamics of the phylogeny, of the epidemiology from the phylogeny. And one of my former postdoc advisors, Saliato, made this really astute remark, is that basically what we've uncovered here is something population genetics have been studying for a very long time, and that's effective population size. Is that in population genetics, we just accepted for so long this complicated term called any effective population size. This doesn't have any natural correspondence to a single consensus population size, but it's basically a number of different processes can lead to the same NE. And so we're basically here, and in population genetics, we just got used to working with NE rather than N. And so there's potential here for getting used to working with these composite parameters rather than the underlying actual epidemiological parameters we're interested in. Maybe we can learn about the dynamics from those. And in a paper studying bacterial diversification, published a couple of years ago, before we knew about all this stuff, well, we actually kind of use these composite parameters by accident to study the diversification of bacteria, and we're able to show that they have signals of these major events through time. So maybe that's a hopeful kind of avenue. But the conclusion from this talk, part of the talk, is that inferences from viral dynamics are generally not robust, and new approaches are desperately needed. Yeah, so macro case, we discovered this. We showed that it actually has borne out in the actual data analysis, and then we kind of extended this to the viral phylodynamic case. And here's the papers I discussed in my talk. You want to write them down, look at them. That's all I have. Thanks for listening.
The likelihoods then just very flat and not, there's just not much information here, so you go.
Yeah, so it's kind of weird because for any given model, like why is the problem so strange? Is for any given model, let's say, oh, we're going to assume a constant birth and death rate or constant birth death sampling rate, there may be a single peak in the likelihood, which says this is the optimum. But there's like a ridge in like meta model space, if you see what I mean. If you can like have these transformations to the model, all of a sudden there's a ridge between all those in that space, which is not normally how we think of modeling. And so this is a really weird problem that, like, at first I started working on this, basically I didn't really believe it made any sense. Because how can this possibly think of this kind of like, well, we're always just so used to, and I'm so used to being trained to like deriving a model, setting a population model, setting consistency, etc., without thinking about alternate model parameterizations that could give the same actual distribution of data. And when you start thinking about that, you actually realize there might be a ton of these transformations that give you the same things. You have like basically a peak in a ridge in like meta model space in some ways. And it turns out really to matter in both these cases, and I suspect in other cases that we haven't realized that much. And kind of, yeah, I'd be very interested in talking about this problem in a more general sense. Yeah, question? Yeah. Oh, yeah,
I guess I was wondering, this might be a little tangential, but related still. I guess we're kind of in this moment of an extinction period, right?
Yeah.
How does, I guess, one, maybe like the incorrectness of these models previously impact what people are saying with that currently? And then if we are going through this extinction period, how might that influence people looking back at the models?
So that's actually a really fun question, or a really sad question. Not fun in terms of extinction's fun, but basically, I've been very curious about this. Okay, so one of the key things we're going through a mass extinction event right now is that it's really inflated compared to what we estimate from the trees. But those estimates for the trees are kind of like unclear. And so I don't really know how to make that comparison. I think there's like a gazillion other lines of evidence that just biodiversity is in trouble. And it don't rely on the rate estimate comparisons for exactly. And I think, yeah, so I think that's a super interesting question. And I've like thought about how to like, I sketched a paper, I wrote a paper that's kind of like highlighted this kind of point, but I don't know how to like think about it really concretely in terms of that. But in some senses, and there's a paper that I was referring to that's come out basically showing that some general features in some cases are preserved across congruence classes. But not all, like different features are not. And so it could be the case, I'm almost certainly the case that like this mass extinction event likely influences the tree in some ways, if you look back on it in a million years, but I'm not really sure exactly. Like, I think that question you asked is like the thing I most want to know, but I don't know.
We don't have the data there.
Yeah, I don't know how to do it. We don't know how to do it, but yeah, Paul.
Yeah, so I mean, I think I'm sort of thinking this the way Jim is, which is, you know, why is this so I mean, I can't help but try to generalize, get a little insight into the way you're thinking about this problem.
Right.
You know, are you thinking about the class of questions, the class of inferences that a phylogenetic tree can't allow you to make? Right?
Yeah.
And so can you sort of generalize from this, like, you know, is there something about the, you know, the birth and death model? Right?
Yeah.
Is it just, if you know, just in a simple little world, right? You know, you've got two rates that you know, you've got a birth rate and a death rate. And if those vary in some, you know, correlated manner,
right,
you're always going to have these equivalence classes.
Yeah. So
congruence classes. So are you talking about classes of problems that you can't make into?
So this is the other elephant in the room.
Yeah.
Is, and I went on a soul-seeking pilgrimage, I would say, after this paper was published, and basically talking to different people. I talked to population geneticists, met these ecologists and stuff like this. And like in some ecology problems, there's in terms like species distribution changes through time. There were actually some of the groups finding something similar, but didn't really realize the connection to our work. Mm-hmm. We also found out like this actually same phenomenon has been discovered in like geomorphology or something like this, basically river dynamics. People have been kind of like this, and this came up in the 80s in some financial models. Okay, so what all these have in common, I don't know. The paper, and I shouldn't say this because this is the paper that there's a huge paper waiting to be written, which like identifies what is the property of the birth death process that we're studying, and the other model that people have touched on that makes them susceptible to these kind of things. And can we identify beforehand whether our model is in that class or not? And like that is a 5,000 citation paper waiting to be written. And I don't want to give away like I hope I write it one day, but maybe one of you can beat me to it.
Yeah, are there any thoughts? Is it like, you know, just a matter of, I mean, not just, but the way I think about it is if, you know, if it's not convex, right, and it's multimodal, you have all these totally different points that lead to, you know, the same height.
Right.
Elsewhere. Right. I mean, yeah, I don't know. Like, it's just, you know, you touch a lot on the geometry, so I'm just wondering, is it through like, you know, the convexity of just the likelihood? Is that been demonstrated?
So yeah, well, I don't know. Like, I mean, like I think that's like a live question. It's ultimately it's like what the geometry of these transformations, why they're all like. I'm not nearly enough good enough at geometry to like my colleague Cillian Luke is like a really good mathematician, and like he was able to prove that these are geometric related. And I think like there is some geometrical approach. And then we were contacted by this guy, this PhD student in Poland who's a mathematician, and he's working on geometry, and he got intrigued in this problem too. And he was kind of like making some headway in the way mathematicians do, which is, in their own abstract universe about how to think about the geometry of these problems. But like, yeah, again, I'm going to speculate wildly by saying there is some general way of thinking about these class of models that might have similar dynamical issues, but I don't know how to find those. But I would bet a lot of money that it's not about a birth-death scenario. It's about birth-death is of a class of models which has these similar kinds of things.
You just need more data.
So that's the thing, you can't just get more data because,
No, I know you can't.
Yeah.
Maybe that's, I know it's all.
right, so I think it's unsolvable because in the sense of you can't just like add more tips to your tree because even if you sample, unless you basically sample so high in your viral phylogeny that like the sampling rate is effectively 100% and that parameter goes away, then you're okay. But I mean, like that's like we're never going to be at that limit. And if you were at that limit, you wouldn't even use these models anyways, basically. But I think it's thinking about the models in a different way and like learning to get inferences from things that we're not comfortable with. The other thing is this other paper that just came out like last week or something like this, working on this problem. And basically what they were able to do was they created a little software piece of like, you could take a given model and it could like basically do what we were doing by like exploring the scenarios, but we were doing it by hand, basically by picking a new view extinction function and then finding the corresponding lambda. But they actually have a way of like sampling model space, of like sampling different parameter combinations and exploring like how wide the inferences are based on like the set of models that they're like sampling this kind of congruent space. And they show kind of like, okay, for some models, to sample the space of inferences are basically all leads to basically the same inference. For other ones, you get very divergent inferences. And they don't really have a good grasp as to why this happens, but at least sampling from the model space allows you to have like some measure of like how confident you are in these estimates, which I think is a good approach as well.
Any other questions here online?
I can maybe I can see here. There's only everyone's ran away.